Our next speaker is Ahmed Kabil. He's a PhD candidate from Dr. Magnagni, and he's going to talk about, Ahmed, a novel transgenic mouse model for innate lymphoid cell lineage tracing and fate mapping. Take it away.
Thank you, Parvin.
Sure, thank you.
Okay, hello everyone. First, thank you to the organizers for allowing me to talk to you today about our newly developed animal models for interrogating the emerging population of innate lymphoid cells. So innate lymphoid cells, what are these cells? Well, to keep it very simple, these cells are the innate counterparts of your T helper cells. They've only been discovered 10 to 15 years ago, so pretty recent. They come in these three flavors, ILC1s, ILC2s, and ILC3s. And these cells pattern the same kind of stereotype modules that your immune system has evolved to interact with the tissues, the outside world, and with organisms. But unlike the T cells, these ILCs do not express an antigen receptor. So the fact that there's this innate group without antigen receptor that is hardwired to make these programs allows us to ask where these cells end up, where do they develop, and whether they could be used as a target in disease treatment. But they're still a relatively new cell type, so there's still a lot of questions on how they are generated. Are they plastic? Can you go from an LC2 to an LC1? These cells tend to be long-lived tissue-resident cells. So are they generated from tissue-resident progenitors, or do they come from the bone marrow or another source? That's still a black box. Are there peripheral populations of ILCs that move around? Are they generated from the bone marrow, or do they come early in life and then be tissue resident thereafter? So there's a lot of questions that are making the field increasingly messy. So we decided to develop some new models and tools to fate map and lineage trace these innate lymphoid cells. So we generated these IL-17RB GFP Cree mice. So using the promoter for IL-17RB, we have an inducible Cree with a GFP. And if we look in the small intestinal lamina propria, we can identify a population of cells that are IL-17RB positive that are lineage negative. So they're non-T cell, non-B cell, non-myeloid cell. They constitutively express CD127 and the IL-25 receptor, which is a selective marker of those ILC2s that are found in your gut. And further flow cytometric analysis showed indeed that these lineage negative IL-17RB positive cells express all of the ILC2 markers, such as KLRG1 and SCA1, but they lack all of the other ILC1 and ILC3 marker. So really consistent with this ILC2 phenotype. This is not just restricted to the gut, it's expressed on all of the ILC2s found across all the mucosal barrier sites of the body, including the lungs, the skin, and even in the lymph nodes. So pretty selective, almost all of the ILC2 populations are marked by this marker. So really a signature of these ILC2s. It's not expressed in any of the other lineages, so it's not on your ILC1s, ILC3s, or natural killer cells. And if you look at all these different sites, it's really only the ILC2s that are getting labeled by this marker. So now to the fun experiments. These inducible Cree mice can be tested using reporter mice. So here the Rosa stop RFP mice, when crossed to the Cree transgenic, if you give these mice tamoxifen, they'll drop out the stop cassette and indelibly mark the cell with red fluorescent protein. And if we look and do this in the gut, all of the lineage negative, IL-17RB positive cells, when you give these mice tamoxifen, they'll light up like gangbusters. Almost all of them are marked by RFP. So clearly this Cree is working as we hoped. Well, what has emerged from the works of several labs is that ILCs develop through a layered ontogeny, seeding pre- and post-birth following an early life expansion. So ILCs that arise from the neonatal period, they seem to persist through adulthood and exist as these tissue-resident cells, which provokes the question of how these cells are self-renewed and whether there are progenitors that can actually contribute to the ILC2 pool found across all these different sites. So indeed, these ILC2s, they come very early in life. If you look in the fetal gut, as they're embryonic day 18 in the mouse, we can identify using our mice these ILC2s in the fetal gut and even in the fetal liver. So they come very early in life. But what has gone under the radar is actually the role of the thymus in ILC development. So it is well known that the thymus is the specialized environment dedicated for the development of T cells. It's like a T cell nursery, if you will. But we actually showed previously that these ILC2s, they have this non-productive T cell receptor gene rearrangements, which actually hints at a thymic origin. And we did a very simple experiment where we did this fate mapping experiment using comparing the ILC2 progenitors in the thymus and the ILC2 progenitors in the bone marrow. And we looked at how rapidly they could turn over. And what was surprising to us is that after 1 day of the tamoxifen scheme, we see beautiful labeling in the ILC2 progenitors, and this gets quickly diluted out after a couple of weeks. There is no more labeling in the thymic ILC2 progenitors. They've left and they're no longer there. In the bone marrow, they don't get diluted, so it's very slow turnover. As you can see here, after 5 days, it stays the same labeling across even after a couple of weeks, really showing that the ILC2 progenitor in the bone marrow, they don't contribute in the adult mouse at least. So everyone thought that ILC2s, they branch off from the thymus and they branch off from the bone marrow and they never grow through the thymus. But their gene rearrangement pattern and the rapid turnover in the thymus really hints at a thymic origin in this population. So with that, I would just like to thank everyone in the lab, mainly Kelly for being a great mentor and making science very fun. Thank you. I hope you enjoyed it.